Hello, I'm Luciano Costa. I'm a myeloma physician at University of Alabama at Birmingham. I had the opportunity to present on behalf of my colleague and co-investigators the study with mezigdomide in novel combinations for relapsed refractory multiple myeloma that we call CA057003 trial. So mezigdomide is a novel, very potent, perhaps the most potent CELMoD agent that has a very enhanced tumoricidal and and immune stimulatory effects that are far superior to the MEZI agents like lenalidomide and pomalidomide that are very well known into our field. In combination with dexamethasone alone, it has some very promising clinical activity, as has been previously demonstrated and published in a population with heavily pretreated multiple myeloma. However, there are several pathways that remain unexplored in multiple myeloma. Some of those pathways that are particularly relevant in late myeloma, they are responsible for faster tumor growth, resistance to therapy, among other negative behaviors of the clone. And some of those pathways are amenable to intervention with therapy, such as the EZH2 pathway, the RAS-RAF-MEK pathway, and the CKS1B amplification that comes from the gain amplification of 1Q. So the study takes mezigdomide, that is a cereblon modulator, and uses a dexamethasone and an agent that interferes with one of those many pro-oncogenic pathways that might be relevant and can enhance the efficacy of mezigdomide by modulating those pathways downstream of cereblon. And this study was based on some very robust preclinical data where we can see that those agents, as respectively tazemetostat as EZH2 inhibitor, BMS 96158 as a BET inhibitor, or trametinib as a MEK inhibitor, are either additive or synergistic to mezigdomide, enhancing the potency of mezigdomide in vitro. So those this study had three different regimens, three different cohorts, and we tested different doses of MEZI between 0.3 and 1, and in some occasions more than one dose of the partner drug. And important to know that all those drugs are oral, so it's an all-oral regimen. Those patients have had relapsed refractory myeloma, multiple prior lines of therapy, and no available existing therapy known to have clinical benefit. Mezigdomide was used for 3 weeks out of 4 in each cycle, and the other drugs were used in a different schedule that has been previously determined. Tazemetostat continuously, 158, 5 days on, 5 days off in the first half of the cycle, and trametinib for the first 21 days of the cycle, very much like mezigdomide. And this is the patient population, 56 patients, median of 6 prior lines of therapy. Those patients had a high proportion of individuals with extramedullary disease. And you have here the majority of patients with ECOG performance status of 1. When you look at prior therapies, everybody has received a PI-IMiD and anti-CD38 monoclonal antibody. The majority of patients, of 82%, had triple-class refractory myeloma. And quite unique for this space, we had the majority of patients, 57%, had received a prior T-cell redirecting therapy, either CAR-T or a bispecific antibody, including more than a quarter of the patients who had disease that had become resistant to a bispecific T-cell engager. And when you look at the different cohorts tested, we find that the mezigdomide and tazemetostat was well tolerated, and all the three doses tested were considered safe and amenable to be explored further. The same happened with trametinib on cohort C, where all doses were tolerated. You had a few dose-limiting toxicities, one on dose level 3, one dose level 4, but that did not cross the safety boundary. On cohort B with the BET inhibitor, we had a total of 5 dose-limiting toxicity events, mostly on dose level 3, and that dose level was considered not tolerated and therefore could not be developed. When you look at other toxicities, they were mostly hematologic, very similar to what we see with mezigdomide and dexamethasone, most frequently being neutropenia. All the other toxicities seems to be a lot less common. Despite the high rate of neutropenia, infections rate was not exceedingly high, with between 15 and 25% of the patients, despite having grade 3 or higher infection, despite their having have had extensive therapy, prior therapy, including classes of therapies that are associated with a high risk of infection, such as the bispecific T-cell engagers. When you look at the efficacy for cohort 1 with tazemetostat, the EZH2 inhibitor, across the 3 cohorts, the response rate was 60%, and you see that the majority of experiences at the 3rd dose level with 1 milligram, and most of those patients had ongoing treatment with a deep response, with a response or a deep response at the time of data cut. With 158, we had an overall 35% response rate, so it seems somewhat lower. The cohort with the higher rate of response was cohort 3 with a higher dose of MEZI, but it was also the cohort that was not tolerated with too many dose-limiting toxicities. For cohort C with the MEK inhibitor trametinib, we see across the board 75% response. I would alert you that the last cohort seems to have a somewhat lower rate of response, but keep in mind that's a very young cohort, most patients being on cycle 2 and cycle 3, and some of those stable disease are likely or have converted into a partial response or better. Important that the median duration of response is 6.5 months, which is quite notable for giving how heavily pretreated this patient population is. When you look at the pharmacodynamics, we saw that we have the biological effect of degradation of violas on the dose proportional matter, and the use of another agent does not seem to interfere with that. The same being seen on the B-cell depletion as a biomarker, a biological pharmacodynamic biomarker, and also on the free light chain, which is a biomarker for efficacy. And the same phenomenon was seen with the MEZI in terms of proliferation or activation of CD8 and CD4 T cells that doesn't seem to be at least negatively affected by the partner drug. So in summary, in this first dose finding study of MEZI associated with those other agents, we saw a promising activity and were able to define one or more dose combinations that were well tolerated. We didn't see any safety signal, any toxicity that had not been previously described either with MEZI or with those other agents. And importantly, the hematologic toxicity seems to be very similar to what we see with MEZI and Dex alone, most commonly being neutropenia that is promptly responsive to the use of growth factor and not parallel by a high rate of infections. We saw the expected effect of MEZI on T cell activation and proliferation not being negatively affected by those partner agents. And again, we saw a dose linear increase in the pharmacodynamic effect in relation to the mezigdomide. So in conclusion, we think those results are quite interesting and provide potentially new combinations of all oral agents that can be helpful in patients and using a mechanism of action that is very independent of the T-cell redirecting therapies that are currently being explored and are likely to be developed further. I'd like to, again, thank the support. This is a BMS-sponsored study, and of course was only conducted thanks to the goodwill and dedication of the study teams and the patients and other participating sites. Thank you very much.